Thank you for having me. I notice I'm the only person representing a company amongst all the academics here. But in my defense, I will say that this work was done at the University of Texas by my group for the last 20 years. And I have now decided to work full-time for this company that we started simply because we felt that it's time to actually make products. So the story of catalytic antibodies has actually been a dream since the time of Linus Pauling. And he proposed that one could make catalytic antibodies by immunization with transition state analogs, or as I'm calling them here, reaction intermediates. Now that story didn't quite work out. No catalytic antibodies were prepared for energetically demanding reactions like peptide bond hydrolysis. And the reason is, of course, that the transition state by definition is so transitory that it falls under the Heisenberg's uncertainty principle, that you cannot pinpoint the exact structure. So there are essentially a lot of different transition states, and it's not possible to make an antibody that recognizes a fixed state of the reactant in order to catalyze reactions. So we sort of accidentally found that nature makes catalytic antibodies. You don't have to engineer them. And we discovered these antibodies as autoantibodies for a long time, maybe about 10 years or so. People thought that these were only these are harmful antibodies. But then came the more profound finding that indeed catabodies have originated. These are ancient molecules, millions of years ago, and they have evolved to recognize in a very specific way toxic substances, both in the environment and also self-antigens that are toxic, like amyloids. And this is encapsulated here, that contrary to the common understanding of antibodies, that these are adaptive molecules, you vaccinate and you get a specific antibody. By Darwinian selection, we already have catalytic antibodies that are rather specific, which is going to be the sort of underlying theme of the rest of my talk. So we are able to find catalytic antibodies we think we are ready for development of catabodies as therapeutic agents. We're able to find these using electrophilic targets. So the principle is that the natural catabody has a nucleophilic site that pairs covalently with an artificial electrophile. It binds covalently. This E is a surrogate, that's the surrogate, the phosphonate group here. And this is a surrogate of a weakly electrophilic carbon of the peptide bond. Specificity comes from binding at the usual binding site that gives rise to high affinity binding here, and then there's covalent bonding. So we use these E targets to pull out the best catabodies that nature can give us from large libraries. And then we also use these E targets to immunize animals and induce, much like Pauling used his transition state analogs, we use electrophilic analogs to immunize animals and induce the synthesis of catabodies. And that essentially is encapsulated here. This is the selection for the catabodies, and here is the vaccination. I should talk about this little picture. This is the metaphorical bomb shown here is simply to underline the fact that this is covalent binding, is a high energy reaction, about 50 times more energetic than conventional non-covalent binding. And the consequences of that energy the excess energy of binding, covalent binding, they can be rather profound, which I'll come back to later. It's a much bigger stimulus. It's a kick in the wherever, or it's a bomb going off in the active site, is the simple point I'd like to make right now. And I'll come back to that point. So our data over the last 20 years has shown that catabodies for targeting of the desired molecule that one wants to remove, they are better than ordinary antibodies in all of these respects. The mechanism, unlike one-to-one reversible binding, catabodies work continuously in a multi-cycle way. So one molecule of catabody can digest, can break literally thousands of molecules of the target. These immune complexes formed by ordinary antibodies, particularly for amyloid disease, they can cause bad effects. There are safety problems that have been noted in clinical trials, for example, with Alzheimer. We think catabodies have a better safety profile than conventional antibodies because of the transient nature of the binding. The immune complexes are not stable, and you don't need these phagocytes for removal of the target. Catabodies remove the target directly without requirement for these inflammatory cells. And efficacy, as I've already said, is better. Here is a table, amyloid dissolution. This happens to be transthyretin amyloid by a catabody. And the potency, the direct removal of the amyloid occurs. An ordinary high-affinity antibody has no effect whatsoever. There is no dissolution of the amyloid, as I say, without phagocytes. Now, this platform, very quickly, is really useful across many targets. And the targets are listed here. This is work both from my lab and from several other groups around the world. And you can see there are examples of age-associated and infectious disease targets, autoantigens, oncology targets. The bottom line is that these selection, the E-target selection and immunization procedures, they can generate catabodies and another species of antibodies that we call irreversible antibodies, simply because they bind the target with high affinity, such high affinity. This is covalent binding, and they are the molecular cousins of catabodies. The only difference between a catalytic antibody and an irreversible antibody is that the latter, irreversible antibody, does not support water attack on the complex, so the reaction gets stuck at the irreversible binding phase. But the point of this slide is that we can target most, if not all, proteins that one wants to using this technology. So I mentioned transthyretin, misfolded transthyretin. I say here it's the singular causative agent for several disease indications in the heart, musculoskeletal system, and the peripheral nervous system. There are other targets that one could consider, including Alzheimer, which I'll come to in a second. But we've chosen our catabody cardizyme as our lead antibody for development, simply because we have the target. It's such a clean correlation that the misfolded transthyretin deposits in these tissues that I have indicated, and removal of the transthyretin, the misfolded amyloid, is then predicted, because it is the sole cause of the disease. The removal is predicted to be therapeutic. So I mentioned that the natural catabodies are nucleophilic. They actually belong to the serine protease class of proteolytic enzymes. the colored residues here are a serine, histidine, and aspartate, very much like serine protease, that are the catalytic triad. This is the variable domain of the light chain, and this is the heavy chain. And here is a picture of our electrophilic transthyretin analog containing the electrophile, the phosphonate, that we stuck onto the misfolded transthyretin, and then we pulled out cardizyme from essentially a B cell library. It's a human antibody. I don't have time to show you all the details, all of the data, so I've summarized the data here. This is effective. It dissolves both synthetic and tissue misfolded transthyretin amyloid. There is no inflammation unlike conventional antibodies. Curiously, again, it's highly specific, and this is entirely Darwinian specificity. It's not something I did not immunize an animal or an organism. So the properly folded transthyretin is not broken at all. Yes. It is depleted. Well, I can hear myself. Maybe I won't. I'll speak better, perhaps. So, and it's much faster than, because we selected the cardizyme from the library, we were able to pull out the best catabody, whereas the average catabody found naturally in human blood is much slower in degrading the misfolded transthyretin. So here are some data. This is the average catabody speed, and that's the catalytic rate, rate of breakage of misfolded transthyretin. And because of the selection, we have considerably, it's about 3 orders of magnitude faster than the average antibody. This is one of the clinical indications, the disease indications due to transthyretin deposition. This is in the lumbar spinal vertebra. The transthyretin deposits there in people who are about 50, 60 years old. Many, or 50% of, I would call them middle-aged people, 50, 60, they have mis-TTR amyloid in the backbone. Carpal tunnel syndrome is also a common outcome of mis-TTR deposition. And as you can see here, by treatment with cardizyme, there is complete removal of this brown amyloid, which is stained with an antibody too that binds to the TTR. The cardizyme treatment completely removes the amyloid from the tissue. Now, TTR is often, the TTR amyloid disease is often called an orphan disease because it's found familially, and Europeans and Asians, the incidence of this familial disease is very small. I wanted to point out that in Africa, on the other hand, the mutant transthyretin that causes the disease, it's much more frequent. 3 to 4% of African Americans have the gene, the mutant TTR gene, and this is mostly from West Africa, and then the African American data are shown here. It's called an orphan indication only because it's less common in amongst, as I said, Europeans and Asians. The heart is affected in the later stages of the disease. As I said earlier, the musculoskeletal system is affected sooner, and the age-associated diseases, which are wild type, not the mutant, these are really big indications. And here are some numbers. The number of people, about 40% of everyone beyond 50 has some degree of pathology due to transthyretin amyloid. So this is a really important target, the transthyretin itself, and it's sort of underappreciated. Recently, the good news is, at least for the mutant form of the disease, Pfizer's tafamidis and Alnylam's and Ionis RNA interference drugs have been approved for polyneuropathy in the familial indications. In the long term, they'll probably also be approved for other indications. But the reason I put up this slide is that these drugs, this one, RNAi, it blocks the synthesis of physiological TTR. That's the way it suppresses the amyloid levels. And tafamidis stops it binds to physiological TTR, and it reduces the conversion of the physiological TTR to the misfolded form. They have the likelihood of appreciable side effects because they are not specific for the misfolded form. They cannot even remove the preformed misfolded TTR amyloid. So we think we'll have an efficacy advantage, although we are somewhat slower than these newly approved drugs. And then we have data on amyloid beta that I wanted to show you. Although Alzheimer is much more complicated, the reason I'm showing the data is, one, because it's important, and second, because it's a precedent for the transthyretin work. I wanted to only point out that it's a multifactorial disease. Neuroinflammation is a common feature. Ordinary antibodies either don't work or they cause problems. They actually cause neuroinflammation, which counteracts the benefit of the removing of the amyloid beta. Now, these data, which because of my limited time, I'm just going to very quickly give you the bottom line. We have Alzheimer's that breaks amyloid beta very rapidly. This is an electron microscopy showing complete disappearance of the amyloid beta plaques. This is in vivo data, intravenous therapy with Alzheimer's and gene therapy with Alzheimer's. Both of them remove brain deposits of amyloid beta. We also have Tauzyme, which can be combined with Alzheimer's for better therapy, we think. The main point I'd like to make is that people have almost given up on treating amyloid beta, removal of amyloid beta, simply because so many trials have been done and the results were not very encouraging. We think catalytic antibodies bring something completely new to the table, and they have hope of success where the conventional approaches failed. One couple of sentences about the vaccine, since it's in my title. We induce catalytic antibody synthesis by immunization with electrophilic GP120. GP120 is a coat protein of HIV. This vaccine candidate can neutralize HIV strains across the world, which is the first. And it works in vivo in a mouse model. But the important point here is that, again, electrophilic vaccination, because of that bomb that I spoke of, the kick in the backside that I spoke of, the high energy binding to the B lymphocytes. These are the events that occur. IgM to IgG class switching is accelerated. Deficient antibody synthesis to uniquely vulnerable HIV epitope is observed. And we get the catalysts and the irreversible antibodies as opposed to the low potency regular antibodies. I'll skip and I'll get to this. So we have this platform technologies, the antibody, the catabody technology, and the vaccine technology. And then we have proof of concept for each one of our lead products. And we are also interested in collaborations to broaden the utility of our platform, both academic and pharma company collaborations for new lead products to pre-specified targets. We can produce catalytic antibodies within a period of about 6 months to 9 months. And now that we are a company, perhaps we'll abbreviate that the time frame. With that, I'll thank you for your attention.
We'll have time for just one short question. So.
I wanted to ask, do you have, well, usually, in experiments, you often add some fusion with a tag for purification or for something like this to your protein of interest. Is it possible with your catabodies to just, you give us the tag, we add it, and we use it for ELISA or similar assays?
The Alzheimer's catabody had the tag that we have snipped out for in vivo studies, but that's how we isolated it from a phage display library. The cardizyme catabody is an intact IgM that we obtained from human B lymphocytes simply by recovering the RNA, the genes for the antibodies, and then cloning them into a cell line. We don't need a tag there. And it's a much smaller library. We can pick up our E-target covalent binding by high throughput flow cytometry. In fact, that part has been published. So that's how we pick out the best catabody using the markers that we've used.
Okay. Well, thank you very much.